Hello friends, welcome back to my channel, and today in this video we are going to study about lymph node. Lymph node is a secondary lymphoid organ. So if you can see the structure of a lymph node, what we can say, it is a bean shaped structure which is encapsulated. Now what it contains? It also has the outer cortex, then the paracortex, and then the inner medulla. Now the outer cortex, it mostly consists of B cells. Along with that, some antigen presenting cells are also present like dendritic cells and macrophages. In the paracortex, it is mostly consists of T cells. And again in the medulla, in the center, it is plasma cells. Plasma cells means these are also B cells, but these are activated B cells that release antibody in the blood. So what we have? The outer cortex, then the middle paracortex and inner medulla. The outer cortex consists of B cells and some antigen presenting cells. Paracortex mainly consists of T cells, and medulla, the activated B cells that secrete antibody in the blood, which we call it as a plasma cells. So now, as you can see here, we have afferent lymphatic vessels from where the antigen enter inside the lymph node. Now, antigen can enter either individually or in association with an antigen presenting cells, because in the cortex also we have some antigen presenting cells. So if they enter individually also, they can get opportunity to encounter with any antigen presenting cells present in the cortex. So the antigen will enter through afferent lymphatic vessels. Once it enter here, the antigen presenting cells including the B cell will encounter the antigen. Now if we talk about the B cells, what it will do is it will engulf the antigen, it will process the antigen, and it will present some part of the antigen on its membrane. Now, from outer cortex, along with this antigen, this B cell will go to paracortex to present the antigen to T cells, because paracortex contain T cells. So, to which T cell it will present the antigen? The Th cells. After presenting the antigen, when the B cell encounter the antigen, this is the partial activation of B cell. When this B cell encounter the antigen as well as with the Th cell, that when the B cell becomes fully activated. So after becomes fully activated, it has two paths. Either it will proceed further and enter medulla, where these B cells are now plasma cells and can secrete antibody into blood, or else it can return back to its position only. Now here, if you can see in the diagram, in the cortex, some center is present, which we call it as a germinal centers. So after interacting with the T cell, the B cells has two paths. Either it can proceed further from paracortex to medulla, or it can return from here to the germinal center. So some population of B cell will proceed further up to the medulla to release antibody in the blood, and some will return to a center. Now this center is called the secondary follicle or the germinal center. Now what this center contains? It contains B cell with antibodies on their surface, but these are with enhanced function, because these are now working as a memory cells, okay, for the next time attack. So these B cells will have the enhanced binding capacity with an antigen. So all these are written here, that lymph nodes, it's a bean shaped structure, consists of stromal cell packed with lymphocytes, macrophages, dendritic cells, and these are the first organized lymphoid structure that encounter antigens. Divided into paracortex, medulla, and cortex. Cortex consists of mostly B cell, macrophages, dendritic cell. Paracortex mostly T cells. Medulla, that contains the plasma cells. Now, if you see the path, whenever any particular antigen enter, they get trapped by the antigen presenting cell present in the cortex, or sometimes they'll enter along with an association with an antigen presenting cell as well. Then it will pass to other antigen presenting cells, including B cells, and then finally to the T cells. And sometimes from this B cell directly also it can pass to the T cell, which I shown you in the flowchart form. Now, T cells in the lymph node. So lymph node contains some fibroblast reticular cells, as you can see here, FRC, fibroblast reticular cell. They are like working as an adhesion thing, like a glue type. So what they do, they will bind, DC means dendritic cells. So they will bind different antigen presenting cell with its surface, and they will guide the T cell because of the release of some cytokine or chemokine. We will learn about cytokines. So chemokines are the type of cytokines only. So we call those cytokines as chemokine that has the property to attract other cell. They worked as a chemoattractant. So because of these chemokines, the T cell will be guided and will reach to those antigen presenting cell with which it can bind. And because these antigen presenting cells are glued with this membrane in the lymph node, so the T cell will get sufficient time to bind with this antigen presenting cells, because these antigen presenting cells contain antigen on their surface. So the T cell should bind with these antigen presenting cells. For that, the T cell should get sufficient time required for proper binding. So because these antigen presenting cells are glued to these membranes, so it is easy for T cell to get that sufficient time to bind with this antigen presenting cells. Now coming to the B cells in the lymph nodes, B cells contact antigens in follicles. This I already explained. The follicular dendritic cells will guide the B cells, also help to maintain follicular or germinal center. So the path I already told you, B cell will contact antigen. That's how it undergo partial activation. It will engulf and process the antigen. Now this processed antigen, it will display on its surface, move to the T cell rich paracortex, encounter CD8 cell and undergoes full activation, and because of which it undergoes proliferation. Now after getting proliferation, it has two paths. Some of the B cells will move to the medulla, becoming the plasma cell that can secrete antibody, and others will return to the follicle, establishing the germinal centers, which we call it as a secondary follicle, which I explained to you in the first slide as well. So what are these germinal centers? These are remarkable substructure that facilitates the generation of B cell with increased receptor affinities because they work as a memory cells. Clear? So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.